Thank you very much for the introduction, and I would like to thank all the organizers for giving me that opportunity to present my data here at that meeting. Thank you for being there and listening. So my talk is focused on tumor biology and the role of the immune system that the immune system plays in the tumor, and of course all the different single cell techniques that we use to analyze these samples. And I'd be happy to take any technical questions that come up to the approach that we chose to do. I'd like to start off with a brief introduction into immune cells that are resident in non-lymphoid tissues, because for a long time, what we as scientists did when we were studying the human immune system is basically look at immune cells in the blood, because they were easily accessible, and it was always thought that they are a good representation of the immune response that is going on. And that is still true to a large extent, but in the last decade, it has become evident that there are a lot of different immune cell types that permanently take residence in non-lymphoid tissues. Some of them are depicted here on this slide, adapted from the review: innate lymphoid cells, NK cells, dendritic cells, and most prominently TRM cells. So TRM cells are basically memory T cells that have taken residence in a given non-lymphoid tissue, and by now there is a large body of evidence that shows that TRM cells are very different to their circulating counterparts, and that they adopt functional and transcriptional profiles that are specific to the tissue that they are residing in. On top of that, TRM cells have been shown to play a role in various biological processes. Of course, they are a first line of defense against infections, be it viruses or bacteria, but they've also been shown to play a role in a variety of autoimmune diseases or inflammatory diseases. And also they've been shown to be a critical component of the immune response against developing cancer cells. So TRM cells are the most well-studied of tissue-resident immune cells, but then there are also dendritic cells, of course, or different types of myeloid antigen-presenting cells, which are a particular interest of mine. And I want to expand a little bit more on that. There are not only dendritic cells, but there are, of course, other cells of the myeloid lineage that are able to phagocytose antigen and present it to T cells. Among them are the macrophages and monocytes, which are typically found in circulation. And if you look at any non-lymphoid tissue at steady state, you will have all these three subsets of myeloid antigen presenting cells present. And I think what is often overlooked is that sometimes people treat these cells as solely being responsible for presenting antigen. But they are not only presenting antigen to T cells, they are also able to secrete a variety of cytokines and chemokines and present co-stimulatory and co-inhibitory molecules. And by doing so, they are playing a pivotal role in steering and differentiating local T cells, and thereby critically influencing the local T cell response in a given tissue. So you can imagine that if you're having any insult to a non-lymphoid tissue, be that a bacterial infection or be that actually a tissue injury, then the function of these antigen-presenting cells will change. They might secrete different cytokines, they might secrete different chemokines, or they might start upregulating certain co-stimulatory molecules. And this will, of course, influence T cell function. So on the very first slide, I mentioned that I want to talk about anti-tumor immune responses, and you might be wondering now why I'm talking so much about general tissue inflammation. Well, if you think about it, when a tumor grows at any given tissue site, there is also a component that is inflammatory. And there has been some great and some very efficient efforts in the past decade to develop immunomodulatory drugs that can boost the immune response against tumors. And I'm sure you're aware of the class of checkpoint inhibitor molecules, which have shown tremendous success in a lot of clinical trials and also now as approved medications. However, in terms of understanding the anti-tumor immune response, we are lacking a critical component, and that is which part of the immune response or which subsets of immune cells that are found in the tumor are driven by the specific tumor microenvironment and not only by a general inflammatory response that would be similar if you were to have, let's say, a tissue injury and a general inflammatory response. And that's basically the question that we try to address in this project is, can we make a comparison between a tumor between immune cells that are infiltrating a tumor and immune cells that are infiltrating a generally inflamed tissue microenvironment, and can we use this to define truly tumor-unique T-cell phenotypes by comparing these two setups. The way we approach this is depicted here on this overview slide. So we had basically access to a pretty unique cohort of human tissue samples in that we were able to collect inflamed oral mucosal tissue samples, which we obtained from people that were otherwise healthy, but were undergoing routine dental surgeries for issues like peri-implantitis or periodontitis, which lead to a local inflammatory response in the tissue. Then we were collecting oral squamous cell carcinoma tumor tissue from patients that were undergoing surgical resections of their tumors. And so that is, of course, not the very same patients, but it's the same tissue site. And for both of these tissue cohorts, we were collecting matched blood for each donor. And then we pursued two parallel single-cell analysis approaches. In the first part of my talk, I'll be focusing on this upper part here, where we basically combined two 30-parameter panels that were centered either on T cells or on myeloid cells, and used a computational analysis approach that has been developed by a group here at the Fred Hutch to identify or to get like a general overview of the different immune infiltrates in these two tissue sources. And then we were in parallel also doing different single-cell transcriptomic approaches. And in the end, the overall goal was to identify tumor-unique crosstalk between APCs and T cells, hoping that this would allow us to identify new therapeutic avenues that might be more specific or maybe more efficient than existing approaches. I'd like to start off with the cytometric profiling, where we first focused on cell types that have been described in the literature as being critical for anti-tumor responses, which are those TRM cells that I mentioned in the introduction. So tissue-resident memory T cells have traditionally been defined by the co-expression of two markers, which are CD69 and CD103, and have also been reported to express PD-1 to some extent. And we were pretty surprised when we observed that when we were comparing our inflamed oral mucosal tissue samples, which throughout my presentation are depicted in orange, to the oral squamous cell carcinoma biopsies, which throughout my presentation will be depicted in red, there was no major difference in the relative abundance of these different CD8 TRM subsets. And more importantly, when we assessed the expression of PD-1, which is a critical co-inhibitory molecule on T cells that is upregulated during inflammation and is actually the target of many immunotherapeutic drugs, we basically found that between these two tissue sources, there was no difference in the expression level of that molecule. We also assessed on a smaller cohort the expression of TCF1, which is a transcription factor that has been implicated in the development of terminally exhausted T cells, which are considered to be a class of T cells found in certain chronic viral infections and also in tumors that through chronic stimulation have lost their capacity for peripheral, for effector function. And again, here we did not see a statistically significant difference between our two different tissue sources. So we're pretty surprised to see this. And thus we wanted to ask or to continue this question and ask which are, if it's not the CD8 T cells, which T cell phenotypes are actually the ones that are unique to the tumor microenvironment relative to a generally inflamed tissue environment. And for this, we utilized a computational method that is termed FAUST, which stands for Full Annotation Using Shape-Constrained Trees. That's a method that has been developed in the lab of Rafael Gotardo, led by Evan Green. And this here is a screenshot taken from the preprint that is up on bioRxiv, and I think their paper is about to be published in a more traditional journal soon. And I don't want to go into all the details here. It's a great paper to read, and in my opinion, it's a really nice method that in our hands worked really well. So basically the way FAUST works, it grows a tree of annotation forest, which is basically like a set of all reasonable gating hierarchies, where it does automatically gate markers based on density distribution and other methods. And then it also has a computational step where it would score how much a given marker would contribute in distinguished in different samples. And then based on that, it would do a statistical comparison between whatever sample cohorts you want to compare and report the phenotypes back, which are the ones that potentially distinguish these two samples the most. So when we did this, the output in a way is like super powerful, but you could also consider it pretty boring because it's basically a list of markers that would define a given phenotype. So all the markers that are not listed here are the ones in our 28 color panel that were considered negative. There were a bunch of CD8 phenotypes, and what is interesting about these is that they were, for the most part, not only PD1 positive, but they showed like co-expression of PD1 and CD38, or PD1 and TIM3, or PD1, granzyme, and CD38. So it's basically indicating what has been actually shown by a lot of other groups, that PD1 alone is insufficient as a marker of exhausted T cells, but you need to have a combination of other checkpoint molecules, like, for example, TIM3, for which there are also clinical trials in the making, I think, at this time. So those were the CD8 T cell phenotypes, but another set of phenotypes that were among the highest scoring ones in the FAUST analysis were actually CD4 phenotypes. And you can immediately recognize that these are TREG phenotypes because they are CD4 positive, CD3 positive, CD25 positive, and negative for CD127, which is not listed here. And then the three highest scoring phenotypes expressed kind of a mix of different molecules. And what we found throughout them was ICOS, which is inducible co-stimulator, and HLA-DR, and PD1, and or TIM3 or CD38. So it seemed that there is a population of TREGs, which is uniquely found in the tumor and not in the generally inflamed tissue environment. We tried to simplify that reported FAUST phenotype for manual gating. And we, of course, first assessed the overall abundance of TREGs. And this is by no means new. As you would expect, there is a really large population of TREGs in most of these tumors, which is shown here in these three flow plots by depicting CD25 versus CD127 expression. And here on the right-hand side, quantified as a frequency within CD4s and as a frequency of total T cells, where you can see that in the chronically inflamed oral mucosa, you have a slight increase compared to peripheral blood, but this is much more drastically increased in the tumor microenvironment. And to make sure that CD125 and 127 is a valuable, or is like a valid way of gating on TREGs in that particular experimental setup, we co-stained in a different panel for FOXP3, CTLA4, CD39, and TIGIT, FOXP3 being kind of the lineage marking transcription factor for TREGs, and CTLA4 being the key immunoinhibitory molecule expressed by TREGs. And as you can see on these red histograms, that the CD25 positive, CD127 negative cells are indeed bona fide TREGs. So that was by no means a surprise, but then we tried just using ICOS and HLA-DR to look at the phenotype of those TREGs. And here in this upper panel, you can see the actual TREG, CD4, 25 positive, 127 negative. And down here, you can see the conventional T helper cells, which would be CD4 positive, CD25 negative cells. And you can see that indeed this ICOS HLA-DR double positive population seems to be enriched in the tumor samples and not really present in the oral mucosa, and of course not in the peripheral blood. And it seems to be only present in the TREGs and not so much in the conventional CD4 T cell compartment. Over here, you can see the quantification of this double positive population, which shows a relative enrichment in the tumor microenvironment relative to the mucosa and the peripheral blood samples, which is not the case for the CD4 non-TREGs. So at this point, we have used our cytometry data and the computational analysis approach FAUST to identify a unique subset of regulatory T cells that seems to be enriched in the human tumor microenvironment. The next question that we asked is which antigen-presenting cells could potentially be interacting with those TREGs or in general with T cells in the tumor microenvironment, and are there any unique phenotypes of antigen-presenting cells? For this, we had a second 30-parameter panel, of which I'm just showing you here. Turn this up here again, of which I'm showing you here an exemplary gating strategy from one of our human tumor infiltrates, where we can identify monocyte macrophage-like cells by CD14 and C11b expression. Here are NK cells, here are lineage-negative HLA-DR positive cells. If we take those, we can identify conventional DCs, plasmacytoid DCs, and within the conventional DCs, the cross-presenting DCs, the CD1c positive DCs, and the double negative cells. And down here, you can see histogram overlays of a set of phenotypic and functional markers for these different subsets of myeloid cells, color-coded here in green, orange, blue, and pink. And what I want to highlight here is that there is considerable heterogeneity of these markers across the different subsets. And some of the markers that we analyzed, like for example, CD206 has in the past often been used as like a M1, M2 macrophage marker. And we could actually see that some of these DC subsets don't express it at all, like for example, the cross-presenting DCs and monocyte macrophage and CD1C positive cells, both of them showed like around 50% expression, and PDCs of course didn't. The same was true for kind of PDL1 and for the co-stimulatory molecules CD86 and CD80, where we also saw largely heterogeneous expression patterns. When we tried to make sense of that data, we realized that our cytometry profiling was not digging deep enough, and that's where the single-cell RNA sequencing came in. And luckily, the previous speaker at this meeting gave a very good introduction to the 10x platform, which is basically what we used here for this approach. Again, we used our same tissue sources. We had like 3 to 4 donors of inflamed oral mucosal tissue sample, 3 to 4 donors of tumor tissue samples, and matched blood. And we basically sort enriched the pan T cell pool, so just lineage negative, CD3 positive, and the pan HLA-DR population to make sure that we are having sufficient numbers of cells in our analysis. For data analysis, we used Harmony, which is in my opinion, great computational tool that allows to correct for batch effects and harmonize the data into one data set. We of course performed cell clustering and DE gene discovery. And one thing that I want to highlight here is that we have really a very large data set here. It's more than 150,000 cells, which allows us to really capture also less abundant subset of antigen presenting cells, for example. For time reasons, I can't show you all of that data, but I want to highlight a couple of things here. So over here, this is basically a UMAP plot where we used a single R to do an automated annotation of the cells in our sample. And without going into the details here, you can see that we basically get a snapshot of all traditionally known T cell and myeloid subsets. There's also a minor population of B cells, which is part of contaminating population from the sort, which is nice that you can still capture that in the single-cell RNA-seq data and exclude it. And over here on the right-hand side, this is probably a more relevant graph where I just colored the cells based on whether they were coming from the blood or from the mucosa or from the tumor. And you can immediately appreciate that for the most part, the blood groups separate from the tissue sources, which is of course not very surprising, but actually the tissue cells for the most part co-mingle throughout this UMAP plot, again suggesting that there is actually a large overlap between a chronically inflamed tissue microenvironment and the tumor microenvironment. And you really need to dig deep in order to find the cells that distinguish those two environments or are unique to the tumor. So we had T cell data in here, and I'll come back to that later. But first, we wanted to focus on the antigen presenting cell population. So we zoomed in on the myeloid population, reclustered the cells and reperformed the dimensionality reduction, and then basically looked at the key differentially expressed genes, which are depicted here in this heat map, and based on that, annotated the cells. Most of the annotation matches what is known in the literature: classical monocytes, non-classical monocytes, CDC2s, plasmacytoid DCs, CDC1s. And then there were 3 things which were kind of notable to us. There was a relatively large population of DC3s, which in the past have often been termed inflammatory DCs, but by now there is a bunch of papers that basically uses the DC3 nomenclature to denote these cells, which you can find over here. They are relatively similar to CDC2s, but express a unique set of molecules that we'll come back to later, and co-express also a little bit of CD14, as you can see here. We also found a surprisingly large population of mast cells, HLA-DR expressing mast cells, which I personally was not aware of before. And then we also found this population up here, which we in the beginning just dubbed tissue DCs, but in the meantime, there has been a publication coming out of the Murad lab in New York, where they dubbed this population MRAG DCs, and throughout this talk, I will adopt the same nomenclature. This is a pretty interesting and unique subset of DCs that is only found in tissues and is marked by high expression of co-stimulatory molecules and certain chemokines, like for example CCL19, as you can see over here. So first, we wanted to make sure that the clusters that we identified were kind of distributed across all the different donors that we had. Over here, you can see the blood, which was mostly made up of classical and non-classical monocytes and CDC2s. And over here, you can see our two different tissue sources, where you can see that for the most part, these populations showed relatively comparable abundance between these two different tissue sources, except the mast cells. Actually, the mast cells were highly enriched in the tumor, and we still need to analyze that in a little more detail. But for the rest of the myeloid populations, the relative abundance was quite comparable. What we did next is we wanted to understand what these cells are actually doing in these tissues, and for that we looked at the expression of like co-stimulatory molecules or chemokines or certain cytokines. And what we observed when you look at the mucosa and the tumor together, you can see that there are very distinct modules of chemokine and cytokine expression. For example, CXCL2, 3, and 8 are primarily expressed by monocytes, CDC2s, and DC3s, while co-stimulatory molecules and, for example, CCL22 and EBI3 are highly expressed by these MREG DCs and not so much by the other cellular population. So it really seems to be that all these different subsets are having very specific functions, which is maybe not too surprising. What we were surprised though is when we looked at the number of differentially expressed genes within each of these populations, comparing the tumor and the mucosa, and we found that there were only two populations that showed a relatively large change in their transcriptional signature. And these were actually the DC3s and the cross-presenting DC1s. These MREG DCs, here I forgot to update the nomenclature. So these are the MREG DCs that have been described by the Mair lab. And we actually don't see that these cells change a whole lot between a generally inflamed tissue and microenvironment and the tumor. So we think they might probably not play such a huge role in the anti-tumor response as one could think. When we looked at the differentially expressed genes that made up this large difference in the DC3s and the CDC1s in the tumor, we found quite some interesting patterns. So these violin plots here are arranged in a way that on the left-hand side, you can see the DC3s, and on the right-hand side, you can see the cross-presenting CDC1s. And the upper panel depicts genes that were upregulated both in the mucosa, depicted in orange, and the tumor, depicted in red, like, for example, CCL4 or CXCL3 or IL-1 beta for the DC3s and CXCL8 or TNF for the CDC1s. And the lower panel depicts genes that were enriched or upregulated in the respective cell population only in the tumor. And for the DC3s, we found this to be the case for CXCL16, and which is the ligand for CXCR6, and TGF-beta1, which is an important immunosuppressive cytokine. For the cross-presenting DCs, we found an upregulation of CXCL9, which is one of the ligands for CXCR3, and IL-18 binding protein, which is a negative regulator or a neutralizing mediator for IL-18 signaling. Having found that there are these distinct changes in the DC3s and the CDC1s, we were wondering whether we could use our data set to kind of link this back to the crosstalk that is found between DCs and T cells. And for this, we used a tool that has been developed by the group of Ian Sayes in Belgium, which is termed NICHENET, which I think is a pretty amazing tool. And I briefly want to walk you through the workflow of this tool. So basically, the cool thing about it is, as an input, you can use single cell data, and you can define whatever cluster you want to define as a sender cell population, and you can define whatever cluster you're interested in as a receiver cell population. Then you can run a DE gene test, so a differential gene expression test on the receiver cell population, and look at differentially expressed genes, and NICHENET will then not only look at the expression of a ligand and a receptor, which would be rather simplistic, but it actually looks at the known expression of downstream target gene networks for a given ligand receptor pair. If, for example, let's say you want to look at CD40 ligand signaling, it's not sufficient to have CD40 and CD40 ligand expressed, but you also need to have downstream signaling genes being active for NICHENET to give it a positive score. And then, as I will show you in the next couple of slides, we validated some of the targets that we found by additional experiments. So here you can see these circus plots, and that might look very overwhelming, but let me walk you through this. So basically on the upper half of the circus plot in red, you can see the receptors on the respective T cell population. And down here, you can see the ligands on the antigen presenting cells that we used as input. And they are color coded by basically blue, which are kind of cytokines, co-receptors. Green, to be honest, that's basically things, I called them other, but where we weren't able to make like a lot of sense of or what these molecules might do in the tumor microenvironment. And in orange, you can see the ones where we found that they were only they were predicted by NICHENET to be only active either in the CD4 non-Tregs, which is this population here, the CD8 T cells, which is this population here, or the CD4 positive Tregs. And we were really stunned when we saw that NICHENET predicted that ICOS ligand, IL-1 beta, and IL-18 and IL-15 are potentially only signaling to the Treg population and not so much to any of the other T cell populations in the tumor. ICOS ligand was probably not too surprising, and neither was IL-15 or IL-18, because it has been described in the literature that Tregs can actually sense IL-18 and IL-15, and that these two cytokines can play a role in either the stabilization of FOXP3 expression or actually a tissue repair program in Tregs. But we were pretty stunned when we saw IL-1 beta coming up, because that's traditionally a more pro-inflammatory cytokine thought to have a role on like effector T cell populations. So we tried to validate this pattern that was predicted by NICHENET, and we started off by looking at expression of IL-1 alpha and beta within the myeloid cells in the tumor. And for that, we just basically isolated the cells and did a brief ex vivo culture in the presence of Brefeldin A. And we could actually see that both the monocyte macrophage-like cells, to a relatively high extent, and to a smaller extent, the DCs in the tumor produced IL-1 alpha and beta. And to assess whether the Tregs can actually sense that, we tried staining for the two known receptors of IL-1, for which there are reagents available. There's the IL-1 receptor type 1 and the IL-1 receptor type 2. And we were pretty stunned when we observed that inside the tumor, and also compared to peripheral blood, it is indeed only the Tregs that express the IL-1 receptor, which you can see here in this histogram in panel D, and over here quantified for a bunch of different donors. So basically around 40% of the Tregs express the IL-1 receptor, and none of the other T cell populations in the tumor or the peripheral blood do it at a detectable level. Down here on these histograms, we compared the IL-1 receptor positive Tregs in red to the IL-1 receptor negative Tregs in gray, and to the conventional CD4s in the tumor in black. And as you can see, the IL-1 receptor positive Tregs are actually the ones that express the highest level of ICOS and HLA-DR, which links back to that population that FAUST discovered in our cytometry data. And these Tregs were actually also the ones that expressed the highest level of IL-18 receptor and the highest level of CXCR6, which was among the targets that NICHENET predicted. So we were pretty excited that we could, for the most part, validate the patterns that NICHENET predicted, and that indeed this IL-1 receptor positive Treg population could potentially be one that is unique to the tumor. So at this point, we found this population, but of course we didn't really know what they are doing, and we weren't really sure whether these are truly a distinct subset of tumor infiltrating Tregs. And that was, so we basically had that finding post-pandemic start. So we had two problems. So one problem was that in the 10x data, IL-1 receptor transcript detection was actually relatively poor relative to our surface protein staining. So on average, I would say 10-fold less than surface protein, which again, by no means is surprising, because we know there's a huge number of dropouts in standard single-cell RNA-seq seq protocols. And also we had, since the pandemic started, we had few and very limited human tumor samples available, and we had to make the call whether we want to do more surface protein profiling or doing sorts for single cell RNA sequencing. So we had to weigh what we could do. The solution that we had here was kind of a lucky coincidence, because just at the beginning of the pandemic, there was the S6 sorter installed here at the hutch. And so what we could actually do is we were able to kind of acquire the very same 28 color data that we used to acquire before on our analytical cytometers. We could basically just use that very same panel, plug it onto our samples for a sort, acquire the data, and in parallel sort the populations that we wanted to sort for our single cell RNA sequencing. And we actually chose here for two reasons to use the targeted transcriptomics workflow, which we kind of made a description of earlier this year, which would not only save us some sequencing costs, but also hopefully deliver a better resolution for some of the targets that we were looking at. And since this is a cytometry interested community, I just want to highlight here that we really didn't do a lot of troubleshooting simply because we didn't have time and we didn't have a lot of samples to play around with. So basically what I did is I just took my target values that I had on my FACS Symphony, threw them onto the S6 and hoped that it would work. And I guess I've been lucky it did work. So I'm showing you here an example where over here is actually a fresh sample that I stained with my standard T-cell panel on the FACS Symphony. You can see the CD4, CD8s, high expression of PD1, some TIM3, the TREGs, on the TREGs expression of ICOS and CD137. And this is the very same donor, but like after cryopreservation and run on the S6 sorter with, for the most part, the same panel. And you can see, due to the scaling, some of the patterns look slightly different. But if you look at the relative frequencies, we were able to basically just port that panel from the Symphony to the S6 and sort in parallel and still have some data that we can use for analysis, which was for us pretty lucky to have. So we did sort various T-cell populations because that's what we were interested in. And in a nutshell, to stay on time, we did targeted transcriptomics run from 3 different donors and focused just on the T-cell compartment. Over here, you can see a UMAP plot where after clustering, you can see the cytotoxic CD8s, the helper T-cells, and then actually 3 different population of Tregs, which over here in this heat map are basically this here, the blood Tregs, and then 2 populations of tumor Tregs. And these actually corresponded to the IL-1 receptor negative tumor Tregs and the IL-1 receptor positive tumor Tregs. Over here, you can actually see expression of IL-1 receptor type 2, IL-2 receptor, FOXP3 highlighted here in the heat map. But since this is sometimes a little hard to see, I'm showing you some violin plots on the next slide, where basically in green, you can see the peripheral blood Tregs. In magenta, you can see the IL-1 receptor negative Tregs from the tumor. And in orange, you can see the IL-1 receptor positive Tregs from the tumor. And all of these, of course, express FOXP3, CTLA4, IL-2 receptor alpha, and ICOS, although there is increased expression of CTLA4 and ICOS and IL-2 receptor alpha in the tumor Tregs. But there were a bunch of genes which were enriched or uniquely expressed in the IL-1 receptor positive tumor Tregs. IL-1 receptor type 2, IL-18 receptor, this here is GITR, that's 41BB, CXCR6, CCR8, which has actually been described by the Rudensky lab. This is CD39, and ID3 is a transcription factor that has been suggested to be driving a tissue residency program in Tregs. And this actually brings me to the end, so that we still have plenty of times for questions if there are any. So I hope I could show you what to us was to some extent surprising is that the immune infiltrate in an inflamed human tissue environment and human tumor tissue environment is actually largely congruent, including T cells with an exhausted phenotype. We could use our cytometry data and computational analysis using FAUST to reveal a tumor-unique subset of regulatory T cells co-expressing HLA-DR and ICOS. Antigen-presenting cells express distinct cytokine and chemokine modules in human tumors, and we could use our comprehensive single-cell RNA-seq data to utilize NICHENET, which suggests that there might be a tumor-unique signaling axis in Tregs via IL-1 receptor. I would like to end with a couple of acknowledgements. So I want to thank the American Association of Immunologists for the Intersect Fellowship, which funded me for the past year, Isaac for the Mary Lou Scholarship, and of course, Martin, who is my PI here at the Fred Hutch, and Jamie, who is a colleague with whom I did many of the single-cell RNA sequencing experiments together, Valentin, Evan, and Raphael, who are collaborators here at the Hutch, and Valentin, who basically taught me everything I know about the analysis of single-cell RNA-seq data. Other members of the Privilege Lab, Doug Dixon, who is our collaborator providing the oral mucosal tissues, Brittany Barber, who helped out with obtaining the tumor tissues, Aaron Tisnick from BD Biosciences for a lot of great discussions and support. And over here is my email address in case you're interested. And thank you all for joining for that talk. And I'd be happy to take any questions. Thank you very much.
Okay, awesome. Thank you so much, Florian. That was, again, an awesome talk, a wonderful talk to end off our first day for MetroFlow. So thank you. And we do have a couple of questions that came through. So we have some time for that, which is great. Our first question from Damien is asking how user-friendly that FAUST analysis is. Is it accessible to researchers with some basic computing analysis, or rather tailored to researchers with advanced training in computational analysis?
Well, that depends what you define as advanced training. I mean, it's basically an R package, which I think is not as straightforward to use for most people. I'm also not like a computational person. For me, it takes some time to just sit down and dig through the vignettes. I think the vignettes are, for the most part, pretty good. So it really depends how willing you are to invest time into it. It's definitely not straightforward in the sense that you can just plug and play and click like in FlowJo, but maybe they might actually write a plugin for it so that you can put it into FlowJo at some point. At this point in time, that's not the case.
Okay. We have another question asking how heterogeneous the tumors are between individuals and what tumor dissociation method was utilized.
That is a great question. So of course, these tumors are quite heterogeneous. We actually had, which is not part of that analysis, we had some lymph nodes as well. And there was quite some difference in the relative abundance of different populations, but in terms of the phenotypes that we found, they were actually pretty comparable, much more so than I thought. And in terms of dissociation, we actually used like a pretty standard method here. We just did collagenase type 2 digestion and followed by mechanical disruption. So we tried to keep it rather short. So we only digest for 30 minutes, not to have any or like too much downstream effect of that. But that worked pretty okay for us, I feel.
Okay, great. And I did kind of have my own question that kind of ties in with that, but I'm sure it's a similar answer, but I can't assume. So in your opinion, do you think that the dissociation of these, of these tissues and the downstream cell sorting has any impact on like potential gene expression? And do you think there might be certain tissues that are sensitive to that, like blood versus mucosa versus, are there some differences across that?
So definitely, I mean, this is actually a great point. I think for the most part, we are just, I mean, everyone in the field is just ignoring that. Of course, if you treat cells for 30 minutes or maybe longer at 37 degrees, a lot of stuff is happening. And I think there are actually some papers coming off, I forgot whether it was from the Broad Institute or from somewhere else, where they looked at signatures that are coming up from that fact that you need to kind of dissociate cells. In our case, I think it's hopefully less of an issue because we're actually comparing tissue cells to tissue cells and not tissue cells to blood that much. So our main comparison is mucosa to tumor, where you would assume, since it's the same dissociation protocol, you have the same kind of artifacts introduced. And I looked at the list of genes that has been published in these papers as kind of artifactual, and we excluded that from the DE gene lists if they came up. But it is a great point. I just think there is no good solution for it right now because mechanical dissociation just doesn't work very well. to really get all the cells out. I think in the end, that's why we might need to do spatial transcriptomics after all, because then you really get like the ex vivo picture. And I'm sure that is the future. But for the time being, I feel pretty confident that what we see is a real biological effect, because we are comparing tissues to tissues. For the cell sorting, I'm actually not worried so much, because cells go at 4 degrees then, and not much is happening at 4 degrees anymore.
Great, thank you. We have a question from Luis asking, or he wants to say it's a very nice presentation. And then for the ICOS positive Tregs, did you correlate with increase in the IL-2 receptor as well as in FOXP3 expression? And is there any idea on this subset level of immunosuppression?
We haven't done any correlations, any formal correlations yet. I definitely see that the IL-1 receptor positive Tregs are also the ones that express ICOS and vice versa. So those two markers do go together. In terms of cell numbers, unfortunately, there's no way that we could do a suppression assay, but what we are working on right now, and we are about to get the data back from the sequencers, we did basically isolate the cells and did short-term ex vivo stimulations followed by single-cell RNA sequencing in order to determine what does the cell do if you stimulate it either with a cytokine or a TCR stimulus. I can't tell you yet what's happening there, as we are awaiting that data coming back from the sequencer, but I hopefully will have an answer soon.
Okay, great. And then I had a question about this high parameter data and automated analysis. So in your opinion, do you think that's the way that kind of we should shift to for the most part, to go to more automated analysis? And then do you think that there should be mandatory QC and validation methods put in place to really check to make sure from a technical perspective that these samples are good to proceed with for automated analysis?
Yeah, I mean, that's a great question. And that's actually a topic that is very dear to my heart. I mean, I'm kind of torn here, because I think if we really want to make the most out of this data, we have to use computational methods. That's just how it is. However, I'm a big proponent and fan of still doing manual gating just to assess data for, like, I call it biological QC, because of course I'm not just looking at dots on the screen, but I'm trying to link that to known biology. And I think you can do a huge deal of QC by just plotting a couple of markers, I don't know, versus T cells, B cells, have you, and just look at the expression patterns that you would expect. Of course, for the automated analysis, you have to have a bunch of QC modules, be that just like a signal over time cleanup and doublet exclusion and what have you. But I think there is no real method yet that would validate the biological meaningfulness and reproducibility of your staining. The way we try to address that is basically we had like one reference PBMC sample, basically like one donor of which we had like a large batch of PBMCs and froze like 100 aliquots. And for each experimental run, I didn't show that in that presentation, we thought one of these reference control samples and stained it along. So we have kind of a longitudinal measure of reproducibility of our staining panel. So I think that would help, and there are actually methods out there, like Sophie made this method where you can utilize that to kind of normalize data, and I think there will be a lot of more developments into that direction, or I hope there will be more developments into that direction. And I think for the time to come, it will still be a mix of like manual and automated. That would be my feeling or my preferred way of doing things.
Same here. And then one more, or actually two more questions. We have one from Michael coming in. It says the presence of the IL-1R on Treg subsets is surprising. Do you think this suggests an antigen independent role for Tregs in tissue?
That could very well be. We don't know that. So we didn't do any TCR sequencing, so I can't tell you anything about the specificity or like clonality of those Tregs. To me, it would be, in the Treg field, I think there are these like two groups of people that say that Tregs have to be antigen specific, and some say they are like more like general anti-inflammatory. I I can't imagine that all the TREGs in the tumor are actually antigen specific. So I would suspect that there is some degree of just like generic cytokine driven suppressive pattern. But I have, of course, no data to support that it's just my feeling based on the literature and kind of what I see in the data. So unfortunately, I can't answer your question. I think that will be exciting to look at in the future. And I could, again, imagine that if we are able to do spatial transcriptomics at higher resolution, maybe combined with like some TCR information, that we would get some answers to these questions, which would be hugely helpful for understanding immunosuppression in the tumor.
Great. And then I just have one more question out of curiosity. Have you tried looking at Hyperfinder to try and, you know, do any of your sorting and trying to, have you found that that's been beneficial? If you have used it, if not, do you have any intentions to try it out?
We actually, that's interesting. We wrote actually a grant, which in the first place got turned down, and then I think other people did that by now. I wanted to use Hyperfinder basically to take single cell RNA sequencing data. So like single cell RNA sequencing, protein sequencing, surface profiling data, and use Hyperfinder to take like transcript defined clusters and find like a way to sort them. That we couldn't get it to work in the end. And I think by now some other people tried doing that. I haven't used Hyperfinder just for like surface protein profiling because we run the 30 parameter panels, and I just used the same panel on the S6. But I heard other people successfully used it to kind of reduce the complexity of their staining panels, and I can totally see that working based on the little playing around with it that I did. But I haven't used it extensively. So.
Okay, cool. All right, I think that's it for questions, unless there's anyone else that has some last minute ones coming through. But it looks like we're all good on questions. So again, thank you so much, Florian, for an amazing talk.
Thank you everyone for joining. Have a good rest of your day.
Yes, thank you everyone. Thank you to our speakers, Vera, Evan, Marcella, and Florian. We really appreciate your time. Thank you to all of our attendees. And I wish we could be in person. We would give you all a round of applause. So on behalf of the attendees, I will do that. And I would like to just share, let me grab my screen. All right, here's again our MetroFlow information. You can find us on Twitter, MetroFlow Cyto, and on YouTube. We have a YouTube channel. If you do go and visit us, please subscribe. If we get over 50 subscribed viewers, we can get a URL that is not this ridiculous string of letters and numbers. So that would be great if you could subscribe. And thank you all for attending. We really appreciate your time, and we have another session tomorrow starting at 10 a.m. We have Tomas Kalina, Nicole Polton, and Rachel Sheridan. So please join us and enjoy the rest of your day. Thank you.